Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Quick note before we begin, the Finding Genius Foundation, as part of the Finding Genius Podcast, has recently completed a book about understanding viruses. So the creation of this book was to interview 100 virologists, ask them a lot of deep, difficult questions, take the most difficult questions, and then re-interview the top 25 or so and ask them the hardest questions I could think of. And we compiled that all into a book. So you'll see question and 4 or 5 experts' answers. Question, 4 or 5 experts' answers. There's about 30 questions in the book. I think it's a great read for the layperson and for the researcher. It talks about a lot of speculation in the world of viruses, such as are they alive or not, and why is it important? Why do viruses go latent or hidden or ineffective or sit in a person or an animal or another creature for weeks, months, years, and then suddenly become virulent and affect that person? So there's a lot of really provocative questions in the book. It's now on Amazon. So if you go to Amazon and type in Finding Genius, you'll see the book on viruses. It's also on Kindle. The Audible version is in production and should be ready in approximately a month. But if you want to go and order it now, you can do so again by going to Amazon or Kindle, or go to findinggeniusfoundation.org and go to publications. There's an opportunity as well to get the transcripts of all the interviews and to hear the original interviews themselves. If we put them all together, the book would be about 1,000 pages, but we condensed them down to make it juicy and concise and tight and very interesting. So I hope you'll check out the book. We're now working on one about cancer, but this is going to be our goal is 3 times a year to come out with these masterclass books that I think will inspire new scientific research, and I hope you'll check it out. Thank you.
Hello, this is Richard Jacobs with the Finding Genius Podcast, part of the Finding Genius Foundation. Today I have Gorin Šimić. He's a full professor of neuroscience and anatomy. He's tenured at the Mediterranean Neuroscience Society. He's their general secretary. So we're going to talk about his research. He's part of the Croatian Institute for Brain Research, CIBR. So Gorin, thank you for coming.
Thank you for the invitation, Richard.
Yeah, if you would, tell me about your research, please.
Well, I started as I worked in a mathematical gymnasium. Then I enrolled in medical school. Afterwards, I worked as general practitioner. Then I went for a PhD at the medical school. And afterwards, I was in the Karolinska in Stockholm for my postgraduate education and also part of my PhD thesis. And since then, I've been working mostly in the field of neurodegenerative diseases. But also, I pursued a number of other projects in brain diseases, in development of the human brain, and some others.
Well, which of your projects do you believe will be most interesting to talk about today? What would you like to focus on?
Well, as you know, significant investments have been given for the Alzheimer's disease research. However, for the last 20 years, there have been no breakthrough medicines. But I believe that in the next 5 to 10 years, there will be significant breakthroughs, and then that we will have some modifying therapies. In my laboratory, we work on several projects. One of them is to find small molecules which will inhibit oligomerization of the tau protein. Tau protein is one of the nasty proteins that accumulates and in the form of neurofibrillary degeneration, and eventually it causes problems with how your brain functions, particularly in elderly people. It is also part of normal aging, and that is one of the major problems, that normal aging processes are, the pathology is overlapping normal brain aging. So it is difficult to find differences, statistically significant differences, if you compare the same age groups. So you have to include younger age groups and older age groups and make some regression analysis to see some significant changes.
Well, what's observed if you look at the the pathology and the histology of brains with Alzheimer's? Do they all look pretty similar, or do they look different in certain ways?
This is so interesting. So since Alzheimer's times, we have those plaques and tangles, but since Alzheimer's times, he also, with others, explained that there are also changes in glial cells and neuroinflammation. And this has been discarded and neglected for over almost 100 years. So today, again, we rediscover that neuroinflammation is so important, and also pathology of glial cells, those supporting cells that are important for metabolism of neurons. So, I was really amazed to see that to read the depth of those people from back 50 or more years ago that were so aware of these changes. Over time, we discovered many underpinnings of these biological processes, and we simply have different interpretations from those times. And it is extremely interesting and amazing how those even pathological criteria for the diagnosis changed over time. Every 4 or 5 or 6 years, you have new consensus criteria. So some criteria early in 80s, when Alzheimer's research started, were totally different from today's. For instance, it was the major criteria was how many, at those times, they were called senile plaques. Today, we call them amyloid plaques. But the main criterion was how many of amyloid plaques you have to have the neuropathological diagnosis of Alzheimer's disease. Today, we know that neither the quantity or volume or distribution of amyloid matters. It has many different biological states. So the diffuse deposits can be seen in normal elderly people who are cognitively perfectly fine. So even PET scan with florbetaben or some other radiopharmaceutical are not indicative of this diagnosis. Moreover, we know that amyloid in its soluble form is protective, is acutely protective mechanism. So, for instance, in traumatic brain injury...
What do you mean it's protective?
Yeah, if you have traumatic brain injury, for example, if you produce more amyloid, then you will get better sooner. So it is a protective mechanism in the short term. But in the long term, it obviously do some harm, and actually it is believed that it starts the reactive phenotype of microglial cells, etc., etc. But in 30 years, the view on those pathological changes has totally changed. It's different, and every day with every new discovery, you have to repaint the whole picture. You have to rethink of what it means and how it goes over time, because from earliest pathological changes till overt clinical picture, it takes 20, 30, maybe 40 years. So one neuropathologist said that neuropathology is when you have 2,000 or 4,000 soldiers on the field, and then each of them shoots to an opponent, and then they all die, and you get the brain on autopsy for neuropathological analysis, and you have to find which one shooted first. And this is difficult because there is, as in all biological systems, you have enormous complexity of players, of those soldiers on the field, on the battlefield.
So Goran, what's different if you look at, again, from a histological point of view, what's different about the brain of someone that has plaques but does not have senility and one that does?
So today, most people believe that senile plaques, or amyloid plaques as we call them today, are just byproduct of what is going on, and that more susceptible and vulnerable neurons, they die much sooner, much before, prior to these typical neuropathological changes. So actually, the more resistant neurons, they sequestrate tau proteins, they sequestrate amyloid. Actually, amyloid accumulates both intracellularly and extracellularly. And then, about 60 to 70% of scientists believe that intracellular amyloid is more harmful than the extracellular.
Before we continue, I've been personally funding the Finding Genius podcast for 4.5 years now, which has led to 2,700 plus interviews of clinicians, researchers, scientists, CEOs, and other amazing people who are working to advance science and improve our lives and our world. Even though this podcast gets 100,000 plus downloads a month, we need your help to reach hundreds of thousands more worldwide. Please visit findinggeniuspodcast.com and click on Support Us. We have 3 levels of membership from $10 to $49 a month, including perks such as the ability to see ahead in our interview calendar and ask questions of upcoming guests, transcripts of podcasts you're interested in, the ability to request specific topics or guests, and more. Visit findinggeniuspodcast.com and click Support Us today. Now back to the show.
So you have processes of buildup and you have process of clearance. Now, in Alzheimer's disease, the process of buildup is based on some signaling. One of the ideas is that beta amyloid serves as a glue to protect blood vessels from breakage, from stroke to occur. So there are special mechanisms in the brain to prevent clot to be formed. And in this way, this amyloid precursor protein, the originating molecule for amyloid, is actually an isoform of protease nexin 2, which is an anticoagulant. So it is believed that this mechanism of oligomerization of amyloid into plaques is over 640 million year old, and it is present even in plants, so in all living creatures. So is
it making the plaques kind of like a biofilm? The plaques, are they alive or are they dead material?
You must have heard of amyloid angiopathy, which means a buildup of amyloid in the blood vessels. And originally in 1984, George Glenner and Kane Wong, they scrapped this amyloid from blood vessels, and they proved that it is the same as in amyloid plaques. Quaranto Vita made antibodies. So it is believed that in one of many theories that there are signals in this neurovascular unit which induce neurons and glial cells to produce amyloid, to fix, to glue potential places where stroke could occur, where breakage of blood vessels could occur. So this signal makes more amyloid. So if you have enough quantities, sufficient quantities of amyloid, you will prevent stroke to occur. Now, stroke is not one disease. It could be thromboischemic, it could be hemorrhagic. It has multiple pathophysiological mechanisms. But it could be the problem that once you start to produce amyloid to prevent stroke, that then there is no negative feedback mechanism to prevent this amyloid to build up, and the problem occurs when these diffuse deposits, when they become insoluble, when they get this form of beta fibrillar sheets. So it becomes stiff, and then it compromises exoplasmic transport, all sorts of things. But
can you have amyloid that is of a different structure, and some of it is permeable and some of it's not?
Yes. So as long as it is soluble, it is highly protective. However, when oligomers form, they may, since last 10 years, we know that it may destroy synapses. It may cause problems with the synaptic transmission. However, the problem is to explain how and why in certain people these diffuse deposits are not cleared. It means that it even has something to do with the lymphatic system, which has been recently discovered, and with the lymphatic vessels in the brain. So since 2 years ago, we know that brain is not immunoprivileged organ, and it has to do with even with your position during the sleep, because this lymphatic system, through aquaporin channels, through blood-brain barrier, it provides efflux of water, and this amyloid buildup on blood vessels, primarily on capillaries, cortical capillaries in Alzheimer's disease.
If you like this podcast,
Please click the link in the description to subscribe and review us on iTunes.
They prevent these osmotic pressures and hydrostatic pressures, so they prevent efflux of water. It seems that clearance is not good enough. Clearance occurs in the slow wave sleep. So as you get older, the slow wave sleep, this part of sleep is getting worse because normally about 43% of metabolites formed during the day, during the activity in neurons that are constantly active, it is being cleared. However, the same neurons which support slow wave sleep, such as dorsal raphe nuclei, serotoninergic nuclei in the brainstem, they accumulate those toxic waste or metabolites, including amyloid and other byproducts of the amyloid precursor proteins and others. So you have a somewhat similar disease, which is called the idiopathic normal pressure hydrocephalus, where you have something different, which is opposite, which is the not the influx, but the efflux, the drainage pathway where these metabolites are not being drained away properly into the venous blood and out of the brain. So in these cases, in one large study, almost 90% of patients have been improved after shunting operation. So if you provide this drainage of metabolites, then you increase the clearance. So as with many other neurodegenerative diseases, you have on the one side you have this efflux and clearance. You have at one side you have metabolism or production of amyloid, and on the other side you have the clearance. So obviously that you have to have a balance between the production and the clearance. So it seems, for instance, that in Alzheimer's disease you have increased production, but the drainage is pretty much okay. In idiopathic normotensive hydrocephalus, you have normal production, but the drainage seems to be compromised. And
one question here: is it a one-way conversion of amyloid plaque from the soluble form to the insoluble form, or can it go back and forth?
Well, that is excellent question, because you always think that there is a point of no return. I believe that there must be at least 2 hit hypotheses, or 3 or more. And many of my colleagues also believe that this second hit could be from divalent ions. So what makes diffuse deposits to become stiff and non-soluble may depend on divalent cations such as iron, copper, and zinc. And for this, it's important to have an impermeable blood-brain barrier. So with aging, you have this kind of increased permeability of the blood-brain barrier. And then, yes, in each amyloid plaque, 70 to 80% of amyloid plaques, you will find iron atom in the center. It might mean that there was the initial point where due to break of the capillaries, some erythrocytes, some red blood cells were spilled and were served as a precipitating center for amyloid plaques to be formed. This is regardless of its importance for the pathophysiology. We just want to explain the mechanistic series of events. But yeah, you would have to take such difficult hypotheses that reach out deep into the evolution are not easily solvable, and you cannot easily check for those. But you collect.
Quick question here. So is the iron bound to a certain molecule, and is it acting as a nucleation site for a plaque?
Yes. So you have in vitro models with cells. If you add a little of small amounts of copper and zinc, you will increase this fibrillization of amyloid for 400 to 500%. So 4 to 5 times quicker this non-harmful soluble amyloid gets into this beta-pleated shape. It becomes fibrillar, and then this process cannot be reversed. It's irreversible. But still, and then it forms those neuritic plaques, which is a combination of neurofibrillary changes and also the changes in the amyloid precursor protein molecules that are product of the metabolism of APP. So you may have heard that there is a group of scientists who believe that there is a primary age-related tauopathy, where these changes occur in absence of amyloid. And also you have a large group of scientists which are devoted to the fact that amyloid is antimicrobial peptide. It was in 2005 when first from this group, LL35, I believe, was discovered. So it has also anti-infectious properties. And it is also crucial in cholesterol metabolism, etc., etc. So when you have any given molecule, say protein, it may be involved at different places, in different processes, in different mechanisms. And there are plenty of them. So it is complexity, the contingencies are endless. So I believe that, human brain cannot absorb all of that. We will be able to handle this problem either with some sort of AI or some machine learning, because it is such a huge amount of data. Only 20 or 30 years ago, we have linear hypothesis. Does amyloid affect this or that process? And then you had only 2 or 3 variables that you were played with, but you were not knowing the whole environment. And that is the problem with transgenic models. In transgenic models, say you have a human gene and you transfect it into an animal, but the product of this gene is an avatar on another planet. So it's over 100 million years of divergence between, say, rodents and humans. So it is, even if you find some way of preventing, for instance, amyloid buildup to occur in vitro or in an in vivo animal model, later on in advanced, in second phase or third phase of clinical studies, clinical trials, over 2,090 clinical trials were actually failed in Alzheimer's disease. So it is.
Quick question here. Since you said that the amyloid plaque contains or is in whole, it's an antimicrobial peptide. Does that point to the fact that
antimicrobial peptide? Yes. So in vitro, if you add this amyloid in vitro, it will in petri dish, it will you have colonies grown on agar, and if you add this A beta, it will prevent growth of staphylococcus and even fungi and many different common bacteria, even viruses. So it has antiviral properties as well. So it has.
Okay, so if this occurs in the brain, would that suggest that there is a microbiome of the brain?
Important.
There's a dysbiosis?
Maybe you've seen this science article last year. It was overstretched. So the results of the study were that scientists found in 6 cases of Alzheimer's disease, they found gingipines, proteins of porphyromonas from the mouth in the brain. And then, the conclusion, the discussion was totally wrong, because either editor or others took that direction, because it was not that porphyromonas caused Alzheimer's disease. It is more likely that those demented people who were demented for years were not having a proper hygiene. So due to the increased permeability of blood-brain barrier, this bacteria, along with its proteins that it produces, enter into the brain. And then during autopsy, you find those bacteria in the brain of Alzheimer's disease patients. So, but it was like the commentaries, the editorials and the comments were like, oh, if I don't brush my teeth regularly, those porphyromonas gingivalis will enter into my brain and cause Alzheimer's. It's not like that. So it is the sequence of events. Most people would like to see the single pathogenetic mechanism, but it seems more likely that there might be many subforms of the disease, and that different molecular pathways can cause common neuropathological changes such as neurofibrillary tangles and amyloid plaques. Tangles could be found in over 35 different disorders and diseases. And amyloid is also a huge group of diseases. So we are talking about just one of the commonest causes of dementia, which is Alzheimer's disease. For Alzheimer's disease, it is defined by both plaques and tangles. So only if you find both, then we can talk about Alzheimer's disease. But there are myriad of others that are underrepresented in one series of several hundred brains. It was 5% Alzheimer's and 6% were Alzheimer's, 5% was Braak's dementia, which is argyrophilic brain disease, for which most of the people do not know much about. So
what do amyloid plaques look like if they're in the brain versus them being in a blood vessel?
That is an excellent question. It is the same molecular structure. It is the same source, but yeah, they behave differently. And since mid-90s, we know that neither volume nor distribution correlate with the dementia severity. However, the number of neurofibrillary tangles and their distribution is positively correlates with the dementia and with the neuropsychological scores or mini-mental or MoCA or other neuropsychological tests. So, and it is also the problem of where it starts. We still don't know where are the initial changes, because today we know that these neurofibrillary changes spread from the site of origin and then to the projecting areas, probably even by retrograde transport, which is a model and experiments we are currently performing in our lab. So we believe we have already some unpublished results that tau changes and tau oligomers could spread by retrograde axoplasmic flow, not only anterograde. And also it is, we saw that activation of microglial cells can occur only due to these pathological tau proteins, even without the presence of amyloid. So all previously dogmatic paradigms were fall apart recently. So glial cells may be the initial drivers. Also changes in cholesterol metabolism, which is also supported by genetic studies.
Quick question here about the plaques themselves. Do they overlap like scales? And how big are they on average? And does that matter at all? What's their shape? What's their size?
Excellent question. Yeah, nobody knows how they grow and why are some larger, why are some smaller. Alzheimer himself noted that some can be seen by naked eye, so they have a diameter of 1 millimeter or so. But my idea, which I published, is that projecting large neurons from the brainstem, which innervate cerebral cortex, from their axons, amyloid can leak, beta amyloid can leak from those axons. And others, Professor Braak and others also have some findings that may support this hypothesis. Because, for instance, serotonergic fibers, they have both synaptic buttons, but they have also what is called volume transmission, which means that they spray their terminal ramifications of axons and the axonal terminals. They release not only serotonin, but also some other molecules. So it is possible that the size of amyloid could depend also on this afferent projecting system. So it could be not only a local production. Since 1991, we know that amyloid that Alzheimer's disease is a systemic disease. Actually, this roots back in 1980 when changes in immunocompetent cells in lymphocytes were found. And then people realized that Alzheimer's disease is a systemic disease. And for instance, fibroblasts in skin of Alzheimer's disease patient, they produce 3 to 4 times more amyloid than skin fibroblasts of controls. So it is the overall production of amyloid beta, but also other products of APP metabolism that are accumulating in the bodies of, and that was the reason why the first vaccination against Alzheimer's disease failed in 2003, because they immunized first 280 patients with the whole molecule, and the C-terminal part of the molecule is highly immunogenic, and it caused meningoencephalitis in 28 patients when this trial needed to be stopped.
Jeez, that's 10%. That's bad, really bad.
Yes.
What were they trying to do? Is get rid of the
first part of the molecule
themselves?
is immunogenic, and it was the so-called peripheral sink hypothesis. Today we know, for instance, since 2003, 2004, we take cerebrospinal fluid and we measure different core and potential biological markers for the disease. So typically in Alzheimer's disease patients, you have decreased concentrations of amyloid beta in cerebrospinal fluid, supposedly because it builds up, it accumulates in the brain. So you have smaller quantities in the cerebrospinal fluid. Later on, in the next phase, you have increase in total tau proteins and phosphorylated tau proteins, which are today accepted as biological markers of the disease. Still, they are characteristic, but not specific. So you have always overlap with controls, with other diseases, and it's always a mosaic of information, and you always play with the reliability and the probability of the correct diagnosis during the lifetime. And then certainly there are many studies that show that biological markers are more informative and more reliable, at least 10 to 15% to clinical diagnosis. And only after autopsy you see that there is a huge overlap of pathologies, and that those comorbidities are general finding. First, you have normal aging, as I said. Recently, a paper was published that each person after the age 48 has neurofibrillary tangles, regardless the pathology. And also after 55, there is no single brain without small infarctions in thalamic territories and elsewhere. And then, you see that some sort of synucleinopathy develops, and then neurofibrillary changes develops, and you have an overlap of these pathologies in various stages. And yes, Alzheimer's disease was present since ancient times, and because evolution and natural selection does not have anything to deal with those wrong letters and changes which occur after the reproduction, in elderly people. So those faulty letters in our genomes, they accumulate over time. So there is nothing that could be solved through evolution. Indeed, more and more variants of different genes accumulate over time, which predispose us for sort of either Alzheimer's disease or frontotemporal dementia, etc., etc. You must have heard that for each genetic change, usually during the evolution, there is some trade-off. So if you get something good during the reproductive period, if some gene product, if some protein serves that you have less fragile bones, it doesn't matter if you later on during senium that you will have some arthritis or something else. So in that sense, it seems that, for instance, tau gene and its variants, it is advised not to call them mutations. So these are all gene variants. So also it is not advisable to say dementia. It is better to say major neurocognitive disorder. But certain processes which we call normal aging, and the sort of processes which are definitely pathology. And Alzheimer's disease is not accelerated aging, but it is a separate pathological process that overlaps, and I missed the term, when it happens, it occurs at the same time, and it seems as normal changes also get worse and accelerate. So but certainly it is a separate process.
Okay.
Only 5 to 10%
let's finish up. We're just at the end.
Are having so-called neuropathologically clear or clean aging. So it is for the first 60 years, you can help to achieve that life expectancy by your diet, by your healthy habits, etc., etc. After 60, from 60 to 80, it is about 50% genetic background, 50% your habits or your dietary regime and your workout and your healthy habits, etc., etc. However, after 80, there is a sigmoid curve. The prevalence and incidence of Alzheimer's disease does not increase anymore. So those gene variants that are involved in pathological processes, they are either present or not. So people with centenarians who live up to their 80s, 90s, or 100 years of age, they have simply good genes. And by your healthy lifestyle, you may increase your life expectancy until 60, 65. But much of what will happen later actually depend on the genes that you got from your parents. So it is highly heritable. So people.
All right. Well, Gorin, we're at the end of time. But what's the best way for people to find out more about your research? Where can they go?
Well, I have this web page, Dementia, HIIM.hr.
Okay.
Yeah.
Well, very good, Gorin. Thank you for coming. I can tell that you're very passionate about this.
My pleasure.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.